Determining gene role in cell morphology of Leptospira biflexa. Leptospira are the cause of the most common zoonotic disease, leptospirosis. Pathogenic leptospires replicate in the renal tubes of their host before being shed into the environment, infecting others that come in contact with that source. Leptospira biflexa, though saprophytic, share a vastly similar genome with pathogenic strains of Leptospira. It is hypothesized that Leptospira biflexa contain genes that influence and determine the development of the morphological structure, as well as the fact that Leptospira biflexa mutant number 2768 contains a mutation that is disruptive in genes responsible for cell morphology. The random transposon mutant library was generated via the conjugation of E. coli strain 2163 and Leptospira biflexa patox strain 1, in which genes are disrupted through the mutagenesis of the transposon containing a kanamycin resistance cassette. Mutant 2768 was identified as a mutant of potential interest when the library was previously screened for observable morphologic and physiologic mutations. Wild-type ATCC 23582 and mutant 2768 were grown in EMJH medium until late logarithmic state. They were screened using a dark field microscope and images were taken through a camera attached to the phototube. The lengths were determined using ImageJ software. Wild-type length average was determined to be 8.706 micrometers, while mutant length average was determined to be 13.7 micrometers. Data was analyzed using GraphPad Prism software, and both datasets were determined to be non-parametric. The p-value of the data was determined to be less than 1/1000th. PCR was performed on purified DNA product for the identification of the gene presence of LAC-Z, kanamycin cassette, and nested PCR product. All three products of each respective PCR reaction came back positive for the presence of the specific gene targeted for amplification. Two samples of an unpurified nested PCR product originally sent out for Sager sequencing did not sequence. When one of them was reran, a sequence was returned. A third sample, purified on-site, was sent out. When it was reran, the sequence shown under the analysis of disrupted gene in LB2768 was shown. All sequences were run through a BLAST program and no significant hints were found. It is possible a spontaneous mutation occurred in lieu of a transposition impacting cell length. Full genome sequencing of mutant 2768 to determine mutation site would need to be done to determine SNPs. Future steps include the DNA purification and nested PCR of a new passage of 2768 to verify findings from original passage, as well as the analysis of new mutants from the library.